Hello everyone, I'm Nepi and I'll be presenting on multiplex CRISPR editing for polygenic or complex diseases. These are conditions such as diabetes, heart disease, and psychiatric disorders caused by many small genetic changes. I'll cover what polygenic diseases are, its current limits, emerging technology, a case study, ethics, and a future outlook. What are polygenic diseases? Most genetic diseases we hear about, like sickle cell or cystic fibrosis, are monogenic, one bad gene. But complex ones are polygenic. They have hundreds of tiny variants across the genome, along with lifestyle factors push someone over the edge into disease. For example, type 2 diabetes involves genes for insulin production and resistance. Heart disease ties to cholesterol pathways. Schizophrenia affects the neural wing, and genetics explains a big chunk, around 30 to 80% heritability, but there's no single fix for this. Current treatment and limitations. Right now, we treat symptoms only. Pills for blood sugar, statins for cholesterol, therapy for psychiatric issues, but we can't fix the genetic roots. Polygenic risk scores, the PRS, help predict who's at higher risk by adding up variants, but they're limited. They're set up to mostly European genomes, so they work poorly for other groups. They give population trends, but not reliable individual calls. And they also miss gene-environment interplay. And they don't lead to targeted fixes. That's where advanced editing comes in. Emerging polygenic editing technologies. The CRISPR-Cas9 revolutionized single gene editing, but for polygenic diseases we need multiplexing, hitting various sites at once. Traditional methods use multiple guides but risk breaks and errors. Newer tools like MOB from UCSD, use orthogonal base editors. Different ones change specific bases safely, no cuts are necessary. This lets us install multiple precise mutations simultaneously, modeling complex diseases better and opening doors to therapy. It's more efficient and less damaging than the old ways. For my case study, I chose the disease schizophrenia. For a specific example, looking at schizophrenia, extremely polygenic with hundreds of variants, researchers used CRISPR to edit multiple gene risk in human stem cell-derived neurons, showing how they disrupt shared pathways like synapses or stress handling. Multiplex editing could theoretically correct several variants to restore balance. It's mostly modeling now, but it highlights how hitting many sites together could have a bigger impact than single fixes. How multiplex editing could help, and some challenges. Multiplex editing could create accurate models or someday lower risk by editing key variants, but challenges do remain. Getting tools into the right cells, especially the brain for psychiatric diseases, avoiding unintended edits, and ethical differences between somatic, which treats one person, and germline, which affects generations. Ethical considerations. Ethics are a big issue. Safety risks like accidental changes or trade-offs, also known as pleiotropy, are real. Who gets access? Who can widen the health gaps? Germline editing raises eugenics fears and consent issues for unborn kids. Many experts call for pauses on heritable edits until safer. Somatic editing for adults feel more ethical today than if you were to use eugenics and create designer babies. Future prospects. Looking ahead, multiplex CRISPR could transform how we handle complex diseases, better models now, maybe targeted therapies later. But we need to diverse data and strict oversight and safety first. It's a powerful frontier with a huge promise if handled responsibly. And here are all of my references.